Hi and welcome. This is June 16th, 2021. I'm Jason Ross with the LaRouche Organization and very happy to be interviewing today Professor Edward Calabrese, a toxicologist at the University of Massachusetts Amherst at the School of Public Health there. Professor Calabrese is a leading world expert on toxicology, on dose responses. He has been brought in to provide testimony to numerous government institutions, industry associations. He's authored countless numbers of journal articles, books. He's a real expert in the field. And he's joining us today to discuss a topic that impacts us all very strongly, whether we know it or not. Namely, how do we estimate the risks of dangerous things around us, things like radiation or substances that might cause cancer, and what mistakes have been made in that field, and how we ought to go about fixing it. So Professor Calabrese, welcome.
Well, thank you very much. Happy to be here.
Thanks for being with us. Take it away.
Okay, thank you very much. Well, the title of my talk, as you can see, is called The Historical Foundations of the Linear Non-Threshold Dose Response for Cancer Risk Assessment. And basically, this is a story, as you'll come to learn, which is really a story of mistakes, deceptions, and failed public policy. It's a real skeleton in the closet of the scientific community and in the regulatory communities that we have today, and one which is which really they don't want to speak about and also don't want to correct. And we'll take a look at how this mistake came to be. And it's not just any mistake; it's a mistake that is affecting major policy, affecting your behavior, affecting your pocketbook, and affecting your lifespan. And so all those things are important to me. So I suspect that that they're important to you as well. Now, in terms of where we go next on this, what we find is that the story of LNT is really a story of personalities, and individuals actually in life can make a difference. And the person who has made the biggest difference when it comes to LNT is a Nobel Prize winner by the name of Hermann Muller. Now, Muller basically...
Professor Calabrese, pardon me, let me ask you if you could, for our viewers, LNT, could you say more about what this hypothesis is?
Yes, LNT stands for the linear non-threshold dose response. And basically, it's a dose response such that when you give a dose of something that's harmful, or it could be something else, and that and every single dose that you give produces an effect, no matter how high or how low. Even when it comes to radiation, inducing an effect at what you might call a single ionization, or with a chemical, a single molecule. And so this is what the LNT model is about. It's saying that essentially, if you're looking at an adverse effect, which Muller would have been doing when it came to the issue of mutations or cancer, it's really saying that there's no escape, actually, that even a single molecule or a single ionization coming into contact, with the body actually is potentially able to initiate the process of mutation and carcinogenesis. And so this...
So this would be in chemical terms, like if a large boulder fell on you, you'd be dead. If a tiny pebble was dropped from a foot above your head, you'd be fine. But the linear non-threshold would say that each pebble dropped on your head is slowly killing you. Is that an analogy?
That's right. And even if you were, and what Muller would say is that if you were hit by a tiny pebble, let's say every hour, on the hour, starting from the time you were born, that essentially the cumulative, like a piggy bank, the cumulative weight of all those, the cumulative force of all those pebbles added up would be equal to if all those petals had been fused together and bopped you on your head at one time. And so Muller would say that the total dose is really what counts. It's not the dose rate that counts. So you could actually take a dose and give it all at one time, or you could give that dose and spread it out in tiny amounts throughout the entire lifespan. And Muller would say those two exposures are equivalent, and that's going to come to the essentially the one of the key battlegrounds in the scientific community is was he right or not? And actually, when he made forth his proclamations, he claimed that it was total dose that was the story. It wasn't dose rate, and essentially there was no exposure that was not without risk. And so that's really where he's coming in from. And Muller, even though I'm coming in from this cancer risk assessment side and this public health issue, back in the early 1900s, when Muller was doing his just starting his career around 1910, 1911 as a graduate student. The big question at the time really wasn't cancer risk assessment, and the EPA hadn't been created and would not be created for another 60 years or so. One of the biggest question that was raging in the minds of these geneticists and other biologists was really trying to come up with a mechanism for the theory of evolution. People were quite aware of Darwin and Mendel's laws, so to speak, when it came to heredity, but they really didn't understand or didn't have a mechanism to explain evolutionary change. And Muller thought that it had to come from changes in the genes, but how did you induce changes in the gene? And so his group of graduate students working under professor at Columbia by the name of Thomas Hunt Morgan, who himself will receive a Nobel Prize in the mid-1930s for understanding essentially the sequence of genes on chromosomes. He basically started his whole graduate group of students to try to be able to induce gene mutations with environmental agents. And they threw everything except the kitchen sink at this little fruit fly that they were working with, and Muller was there for 6 or 7 years, and they used all kinds of ionizing radiation. They used every harmful chemical that you could imagine. They used them at high doses, and one of the most interesting things is, try as they may, and they tried very hard, and not one time, and this is really hard to believe, not one time did these brainy people with what's become a standard model were they actually able to induce gene mutation. If they could have, they would have. And so what that meant to them was that, the genome, it must be very stable, must be very protected. It's really actually pretty hard to induce these gene mutations. And so what happened was that, so that was in from let's say 1912, 13, 14, 15. Muller graduates, gets his PhD, ultimately makes it to the University of Texas down in Austin, and he's you might say manipulated, changed his model to try to make it more sensitive, and finds a way to do this, and then in the course of some experiments in his lab in November of 1926, Muller is able to induce, to his shock and surprise and wonder, he's able to induce massive, what I call phenotypic or visual changes in the next generation of fruit flies. That's called really a phenotypic or visual change in a characteristic. And when he saw that, he concluded that he had induced tiny mutations, and he introduced the term point mutation, even though they didn't know what the structure of a gene is that we might know today. But he introduced this term point mutation, which we use quite commonly today as, for kind of the smallest genetic change that you could possibly make. And so he basically was in a race with others to be the first, because he knew that whoever discovered gene mutation was likely to get the big prize, which was going to be the Nobel Prize. And so he, as I said, he was going hard for it. Now, one of the interesting things here is that this was like a big breakthrough. So when Mueller attempts to publish his big breakthrough, and he does publish it now in July 22nd of 1927, in the biggest journal in the world, which is a journal called Science, that everybody tries to get their papers in. You really try first if you think you get something hot in the journal Science and the journal Nature. And because they've got such history behind them, such publicity behind them, the media follows them very, very carefully, and it's extremely big. And so basically, as I got interested in the Mueller story many years ago, I went back to that Science paper, that July 22nd, 1927 Science paper, where Mueller essentially, I thought, laid it all out and showed all the evidence that he had for gene mutation. Now, you'll be surprised, and maybe you'll be as disappointed as I was, that in this most momentous publication, in this big-time journal, the biggest journal, most prestigious, hardest to get published in, really, what happened is that Mueller didn't show any data in his paper. So he has this most important discovery. He claims that he has discovered gene mutation, and yet when he actually presents or publishes this paper, he shows no data. So you might ask yourself, how the heck did Mueller get his article published in the journal Science and not show any data? Now, I can tell you for sure that wouldn't happen to me. I've tried to get in Science plenty of times with lots of good data. And basically, they oftentimes won't even send it out for review. Mueller, with no data, gets his paper in the most prestigious journal in the world. So how does he do that? And I'll tell you, that's been, a mystery that's still unresolved. I have looked at this in great detail. A little behind the scene story is, at that particular point in the history of the journal Science, it was actually not owned by the American Association for the Advancement of Science, what they call the AAAS, which is this big organization of scientists, nonprofit group in the country. It was actually owned by an individual, and that individual, for about 25 years, was a professor at Columbia University. Just what? The same university that Mueller had been a PhD student at. And while this gentleman, this editor of Science, was at Columbia, he was very close friends with, guess who? Mueller's PhD advisor, Thomas Hunt Morgan. Oh, we begin to see the connection here between Mueller and Thomas Hunt Morgan, and Mueller and Morgan got several articles published in Science when he was a graduate student, and they undoubtedly had some communication with the editor-in-chief of Science, and so Mueller sustained that relationship. And so when it came time to get his stuff published with this most important circumstance, how did he work the deal? And I have to tell you, I have really looked into this. I have obtained Mueller's original. I have a copy of Mueller's you might say Nobel Prize research notebook right behind me in this office, and I have tracked the language and the time of the article to this notebook, and Mueller did actually three experiments that ultimately led to his getting this Nobel Prize. And I could tell from tracking language and how he described things that what Mueller actually discussed in his Science paper, I believe, was only experiment one, which may be why he didn't want to show all the data. And then he subsequently completed experiment 2 and then experiment 3, and but yet he gave how he weaved and told the story. He made sure he wanted to have primacy, or being first, because being first means you get the award. Okay, being first. He knew that there were about 2 or 3 other laboratories in the country and maybe outside the country that were closing in on him. He didn't know how close they were. The people hold their cards close to their vest. They don't want the competitor to see it. The closest one to him was a Louis Statler at the University of Missouri, a plant corn geneticist, radiation geneticist. And as it turns out, he's about 3 months behind Muller. He's breathing hot down Muller's back, but Muller doesn't know exactly how close. So what Muller does is he tries to, you might say, get around the rules, get around, protocol, play a little dirty, use a connection. And he does. And he gets in 3 months early, and he doesn't have to show his data. And there's nothing for a reviewer to actually even criticize because there's no data to criticize. There's no study designed to criticize. Muller actually broke protocol, began to show the kind of person he really was. What was more important to Muller? Playing fair, playing by the rules? Everybody else would have to go through peer review. Did Muller have to go through peer review? No, he used a trump card, and he used it. It's like a get out of jail free card. And guess what? That little move, deft move by Muller, guess what it did for him? It got him a Nobel Prize. Because if Muller he knew that if he had to go through peer review, well, you never can tell what's going to happen in peer review. You just never know. It's sent out to multiple people. They can hold you up for weeks, months, longer than that. They can tell you to go back in and do more research. He didn't want to hear any of that stuff. He wanted to do what? He wanted to be first, and he became first. So what actually happened is that but he ultimately had to show his stuff, right? And so what happened next is that that was publishes his paper on July 22nd, 1927. He gets invited to or goes to a very large, the 5th International Genetics Congress in Berlin, which was September 11th through the 18th. So Muller
excuse me, let me just add, just to clarify again on what this paper showed. This was the first published paper that made the claim that small amounts of radiation could cause point mutations, and that he showed that for a fact. Am I getting that right?
Well, mostly right. What I'm going to say is, because you inserted the word small levels or small exposures of mutation, whatever doses Muller used, and we will look at the issue of small versus large and what is large, whatever doses that he used, he induced tiny mutations. Okay, he induced what he called gene mutations as compared to blowing big holes in chromosomes. In the past, before Muller's paper, other people had induced genetic changes, and you could see some of these phenotypic changes in subsequent generations. You could do this, but it was always believed that those people had just poked gigantic holes in the chromosomes recombined, the genes somehow were the animals, the offspring survived, passed on these traits to the next generation, and you could see changes. And people said that's not really how evolution worked. Evolution worked, they thought, by small micro changes.
I see. So he was claiming that he had experimentally created those small changes, like maybe point mutations that were believed to be characteristic of evolutionary change.
Exactly. That was his assumption. That was his belief. And in fact, he entitles his article, the artificial transmutation of the gene. He emphasizes the gene. That was really very significant for him. Now, I'll get back to the other comment that you made when you mentioned that he induced these changes with small levels of small exposure. I've gone back and I have calculated the extent, the amount of exposure that he gave relative to the background exposure that we're currently receiving right now, more or less, okay, and that we're, that his control groups probably did back in the day, 1927, because the background radiation really doesn't change a whole heck of a lot. It's coming in from cosmic exposures from the universe and some from crustal Earth and so forth. And so, but the dose that Muller gave, his low dose that induced mutations in this first study, just happened to be 95 million times, million with 6 zeros after the 95 million times background. So Muller actually induced very tiny changes, he claimed, with a gigantic dose. And that dose, these little fruit flies, they tend to be very resistant to radiation. But most of the males that he exposed at these high doses, they basically became sterile. And so he was causing some major changes in his animal model. But still, Muller made his claim. Now what happened with Muller is that he makes his claim, but he's got to show the goods. So he goes over to Berlin, and in September, basically about 1,000, 1,200 people show up, all geneticists waiting to hear, can he deliver? And Muller delivers. He shows his data for the very first time, the results from the 3 different experiments. I'd have to tell you that he didn't, his write-up and so forth, he claims that he didn't have enough time to finish the 3rd experiment and didn't include a control group. It just kind of shows how life was working at that particular time. And but he shows the goods, he shows his changes, he convinces the world, and by the time he makes it back from Europe back to Texas, he's a world conqueror. He is now a major player in the scientific community, the first person to induce gene mutation. It's a big time situation. However, Muller has a very close friend by the name of Alan Altenberg, and what he does is he basically challenges his best friend, who's Muller, and he says, how do you know that when you induce these gene mutations that you weren't just like using like a bazooka, knocking big holes in the chromosomes? He said, how do you know? You don't have the proof for this. You just because you see this change in the color of the eye or the length of the insect's antenna or changes in the wings, you don't have any idea at the chromosome level what's actually going on. And so Muller basically took what his friend told him under, you might say, advisement, so to speak, and Muller was concerned because Muller made the claim that it was artificial transmutation of the gene. And so Muller knew that he was going to be in trouble for that assertion. And I believe that was one reason why Muller attempted to avoid showing the data initially and peer review. And I have gotten the publication of that presentation that he gave in Europe. And the paper, if you were to read it in today's eyes, but even in the eyes of probably 1927, 1928, it was a terrible paper. And why was this Nobel Prize research a terrible paper? Well, in the world of science, we structure papers in a certain way, but every research paper that does experimental work has to have what they call a methods and materials section. And that tells you exactly how the study was done, what was measured, how you measured it, where you bought the chemicals from, everything, because people have to be able to go in and to reproduce the work. And that tells you exactly how. And then you have to discuss your findings with other literature that's out in the field and how does it fit in and why. In Muller's paper, if you go back into it, you will find, guess what? That Muller shows and provides no section on methods and materials, none. And he provides no references at all in his article, discusses nothing that's out there in the literature in relationship to his own. So just from that point alone, if Muller had sent that manuscript out for a peer review to any journal, really, it would have been sent back to him and said, we're not going to review this until you actually do it right. And so why did Muller do this? And why didn't he clean it up? Because this is his Nobel Prize work. And the paper that I am just describing to you is the paper that contained the data in his tables and so forth. That was the published real version that became the basis for his Nobel Prize. I knew that this paper could never have been peer reviewed because of the inadequacies that I just mentioned. And so lo and behold, as a result of all my little historical research into the lives of Muller and everything he touched, this is hard to believe too, but I bought 2,000 letters or pieces of communication between Muller and this Edgar Altenberg, his best friend. And in the course of one afternoon, a couple years ago, I just happened to be reading a July 8th, 1946 letter. Now, 19 years after he discovers this thing. And in that letter, he's asked some sort of a question that brings him back in time. And wouldn't you know, Mueller absolutely admits that the paper that he read at that Berlin conference back in September of 1927 was printed or published exactly as read, in Mueller's own words. So I had, wow, proof positive to me that that Mueller actually, it was never peer reviewed at all. It was pretty much a joke. And so, so what happens is that if you go into the big database called Web of Science, where they tell you, which papers are, how many times they're cited and so forth, the paper that gets cited by all the scientists who go back to Mueller, they cite the science publication because it's the easiest one to find and so forth. But it's a publication that doesn't contain any data, none. I'm sure I cited Mueller's paper on different occasions, never really went back and got it. But if I had, I'd have found that there was no data in it to support his conclusions. So this is the man we're looking at here. This is the guy who's going to play a very significant role in the whole belief system for LNT. Why is it going to? Because the whole idea of cancer was predicated upon the assumption that it could be that its underlying mechanism was mutation. That's why it all goes back to Mueller. And it's also going to go back to Mueller for the nature of the dose response as we get into it. So basically what happened is that Mueller, he was first, he won the prize, so to speak, but his best friend told him, you better watch out because you made the claim that it's point mutations. You're going to have to prove this. And ultimately what's going to happen is that a bunch of people begin to challenge Mueller. And what's going to be shown is that if you were to fast forward to really the late 1980s and into the 1990s, when we had really good advances on nucleotide or gene sequencing and understanding how genes are constructed, and people went back in and they actually did expose the animals to the level of radiation that Mueller exposed his fruit flies to. And guess what they found? They found that Mueller actually, did he induce point mutations? No. What he did was he did the equivalent of shooting those chromosomes with a bazooka. He induced massive gene deletions. I mean, holes. And by now, how big? If you look at a single nucleotide in a setting that could ultimately be part of a sequence of what the DNA is. Now, some of these gigantic nucleotide gene deletions, they could knock out hundreds of thousands of these nucleotides all at one time. Boom, big hole. And Mueller's talking about little tiny changes. Mueller had no clue what he was doing, but guess what? Had no clue, but he was totally confident, and he was the smartest guy in the room. And so, as it turns out, well after he's dead, he's ultimately proven wrong. And even his former students and so forth, they admit in writing that Mueller actually got it wrong. Now, I'd have to tell you that Mueller actually 20 years later, 19 years later, in December of 1946, Mueller gets the Nobel Prize for what? For being the first to induce gene mutation. So, but I think you've just heard that Mueller actually didn't induce gene mutation. So Mueller gets the Nobel Prize for something that he actually didn't discover. So that's how this story starts off, fame and glory.
And so this I guess we're going to get into what the impacts of this had been, but this was important to us today because you said that this was to help support the view that cancer is caused by point mutations or small mutations, that this is what the cause of cancer is, and that this is related to this research.
That's right. that's ultimately what's going to happen is that our major regulatory agency in this country, the U.S. Environmental Protection Agency, they have based their whole cancer policy when it was first created in the mid 1970s. They based it upon the assumption that whether it was coming from radiation or from chemicals, both being carcinogens in this particular case, that the mechanism was induced mutation, and it was gene mutation, and essentially in the years, a few years before that, the very famous geneticist Bruce Ames had several papers saying carcinogens are mutagens and mutagens are carcinogens. This became mesmerizing to the new generation, I'd call it the Rachel Carson generation of environmental movement in the 1970s, really focusing on mutation as the basis for cancer, and ultimately becomes the basis for this linear dose response and cancer risk assessment. And it all goes back to Muller, but you might be wondering here, how does Muller relate it to dose response? Well, what actually happens is that Muller does relate it to dose response very early. Now Muller's own Nobel Prize work was not really designed to take a look at the dose response. So what he did was he got two different students working in his lab to use extremely high doses, and they had 4 doses in their respective studies, and they showed a linear dose response at extremely high doses. Now, they were also in this bazooka land of high dose and just popping holes in chromosomes, and they were not inducing gene mutations themselves either. But they saw a linear dose response relationship between the exposure and what I'm going to be calling are these phenotypic changes in the next generation. And so Muller looked at these two students' findings, and he then makes a very big leap. And this is the leap that we're jumping into today in this presentation. He creates a term called the proportionality rule, a proportionality dose response. Proportionality is just another term for linear dose response relationship. He creates the term, it ultimately gets changed to linear dose response in a number of years, but it all started in the literature in 1930 when Muller created the term. Now, I have to tell you that Muller didn't come out of this battle unscathed, because what happened is that scientists challenge other scientists, and what happened is that there was a lady by the name of Barbara McClintock who will ultimately win a Nobel Prize herself, but she was a geneticist, and she would do work at the cell level and do staining of cells. And what she did was and she was working with corn. And so she linked up with this guy, Statler, who was Muller's scientific rival. Now, Statler had published a paper in the journal Science confirming Muller's work, but with corn, showing that high doses of radiation could induce, quote unquote, gene mutation in the corn as well. But he agreed. Muller beat him, so Muller gets the prize. But then Barbara McClintock out in Columbia, Missouri, begins to link up with Statler, and Statler wants her to apply her new techniques for how she can have much greater resolution in looking at what's going on at the level of the chromosome than ever could be done before, and she was the first one, very well known for this. And so but when she looks at the chromosomes when they're really in a very elongated state within the reproductive process, what she finds, and she can see this with much greater resolution, she sees that when Statler exposes the corn cells to his x-rays, and he sees changes in the next generation, he's saying this is due to a gene mutation. She looks at those cells with her new fine eye, and she says, you're not inducing tiny point mutations, like Muller said, you are knocking big holes in those chromosomes, and I can prove it to you. And so she showed him, and he got very, very angry, very defensive. He wouldn't accept it. And then eventually, she hung in and didn't back down. And ultimately, within a month or so, she basically convinced him that his interpretation was wrong, and that when he published his paper on corn in the Journal of Science, that his interpretation was wrong. He had not induced gene mutation. So guess what that makes him begin to think? He said, well, heck, if I'm wrong, guess who's also wrong? Muller's wrong. So I obtained letters from this guy with other people in which he's feeling out their opinions on his new interpretation, and he's getting positive feedback that they think he's right. And so now in 1932, the sixth International Genetics Congress, the one that 5 years after the one that was held in Berlin, this one is held in upstate New York or mid-state New York at Cornell University. And because Muller had become famous, and Statler was also pretty famous, pretty much equal to Muller, they were invited to give what's called plenary addresses or major addresses at the opening of this big Congress. Now, you got to picture this situation, right? Just imagine that you are Muller. You are very big in the world of science, right? And people you don't really know what Statler or anybody's going to say. Statler goes up there and Statler decides to draw a line in the sand, right? A line in the sand. And he goes and he claims that Muller was wrong. He didn't induce gene mutation, that all he did was essentially blow out big holes in his chromosomes, and he deluded himself. And he showed the data of his own data and the work of Barbara McClintock right in front of this audience, a full audience of this international group at Cornell University. It actually totally wiped out Muller mentally at this conference. He was rattled. Became quickly disheveled. You almost have to feel sorry for the guy, right? That somebody just blew him out of the water in a very, very highly visible situation. Now, to add insult to injury, Muller was also going through a big time divorce. It was an angry one, and he was under great stress from the divorce. Great stress that Statler put him under, and at the same time, Muller was involved with a undercover subversive Soviet student society or group, and the University of Texas was really opposed to Muller's advising them. And so what happened? They wanted to get rid of Muller. And so if you take a look at how much pressure can somebody take, his professional world was falling in, his personal life was falling in, and the university that one time thought he was great is trying to get rid of him. And so what does this mean? What it means is that Muller tried to commit suicide at that time. And thank goodness Muller wasn't too great with figuring out doses, right? Because what he actually did was he used the wrong dose when he tried to kill himself. He used a dose that essentially knocked him out for a number of hours, but he woke up and was groggy in an agricultural field in Austin, Texas. And when he didn't show up for work that morning, faculty member and a couple of grad students went out and they found him staggering around, and he had written a suicide note to Altenberg, and not to his wife, but to Altenberg. And so what happened is that Muller, as you can imagine, just had to get away. And so he got funding for a Fulbright fellowship to go to Berlin. And he goes to study and to work with some very, very famous Russian who's now in Berlin, this guy, Timofey Rizovsky, who is, it's kind of like Muller's equivalent, but he's a European. And but during this time period, Rizovsky had linked up with a couple of big time physicists, and they would try to look at this interface between genetics and physics. And between the three people, Dr. Zimmer and Delbruck, who would ultimately win a Nobel Prize himself, They took Muller's data, and which was a dose-response relationship, but they gave it something that it didn't have before. They gave it a mechanism. This was very significant, and they based this mechanism on target theory. They tried to estimate how big a gene was and how much the damage was, and built something up mathematically so that they could try to describe how many hits it would take to, and how this would affect the shape of the dose response. And a single hit, if it worked with a single hit, would come up with a beautiful linear dose response. And so they published this paper in 1935, and it basically was the first time we had a single hit linear dose response model. That's where it all comes from, this 1935 paper. Now, the problem with this paper, even though it had some smart guys to it and nice theory, the problem with the model is that it was wrong from the start. It actually was based on Muller's incorrect gene mutation conclusion. They all assumed that it was that they were modeling a point mutation, when what they were really modeling was a bazooka mediated blowout of large deletions. So what actually happens then over the course of time is that, as we go back to this 1932 blowout at Cornell between Statler and Muller, it created a who's right and who's wrong here. Muller's got pride. Muller's a smart man. He's a hardworking man, and he's very achieved. And the same is true for Statler. They were at the top of their game at the right age, and they had it can only be viewed as a heavyweight championship bout between these two, except it went on for 25 years until Statler died, 20 years. Statler dies of cancer in 1954. And between the two of them, they tried to see who was right on this mutation thing. And I got all their papers. I actually even got their approved grant proposals. I read everything about these people. Muller is really the most he can be obnoxious, he's pushy, he's argumentative, he's aggressive as all can be. Statler's reserved and quiet. He's the dignified man, really. And between the two of them, they basically, and with a high level of professionalism actually between the two, they duked it out for year after year. And I watched all their papers, and it was kind of like when you watch a chess match between two people that are really quite good, but one is slightly better. And the one that's slightly better always, somehow each move is a little bit better, the thought process a little clearer. And after a number of moves, eventually the degrees of freedom are greatly reduced, and the one who's not doing so well and is backed into a corner and ultimately sacrifices his king and dies. That's what actually was happening between Muller and Statler, is that Statler had really shown that Muller's alternative explanation to try to think about how he had evidence to support that there was gene mutation was just about, it was so fragmentary, it was not a sustainable argument. Now Muller's as smart as Statler. He may not quite think quite as clearly, but if they're both at a very high level here, nobody's probably as good as either one of them. But they were the tops of the group probably at the time. And so what happened is that Muller knew that, he was dead when Statler was killing him. And so Muller knew that it wasn't going to work if he continued along the same line. So while in Europe, Muller came up with a new approach. And at this point in time, Muller had, you might say, migrated and was supported by the Rockefeller Foundation in an appointment at the University of Edinburgh in Scotland. And he got a graduate student from India to work with him. And they were going to go back to the question that you asked me right at the beginning of this presentation. And that was this total dose versus dose rate. Muller felt that he could kind of throw Statler off his back if he could answer this question. And that is, if he took a high dose of radiation, exposed it to the fruit flies, and then he took that same dose of radiation and he spread it out over the entire lifespan of the fruit fly, Muller predicted that the amount of genetic damage should be the same. And that would, if that was true, it would be an affirmation of the piggy bank model, you might say, and a total dose interpretation, because Muller believed that every single dose was harmful. The doses were cumulative. You could not repair them. And if you added all that together, essentially you got a linear dose response relationship. The dose rate issue, that was actually saying you could experience some level of exposure, but then if it wasn't too high, you could kind of repair that exposure and then just wait for the next one, the next one, the next one. Muller says you never repair anything. It just adds up like pennies in a piggy bank. And so he basically got this student to do this research where they give it all at one time or they spread it all out. And without going into all the details of that, the student came back after X number of months, and the student showed data that actually supported Muller's interpretation. Muller was happy based on the letters I read, because I got all the letters in between Muller and the student. I got the dissertation. I got all the comments from the dissertation committee. See the whole picture here actually, and so this became a very significant thing. However, I'd have to tell you that I have gone into this everything on what went on to the best that I can figure out, and that the student actually did not do a good job in this dissertation. The study design had numerous flaws. It had execution flaws. He wasn't particularly good at doing these technical crosses. I got all these letters between him and Muller. Muller was not even there to advise him much at all. He was traveling quite a bit, and I have. He changed his insect model halfway through. He didn't calibrate his doses well. The temperature control was poor. He did work that would be mostly hypothesis generation, and it really probably shouldn't have even been accepted as a dissertation. But Muller got the result he wanted. He got and basically did cover for this guy. And really, and I've written some, and I even believe that both he and Muller hid data that didn't that wasn't supportive of their interpretation, and I have good documentation for that. In any case, what Muller did was make the claim that in fact this was very important, that this guy was right, and it was this total dose explained this, and that you could take this total dose and make an argument that that his work was actually being able to induce mutation with a very low exposure. So he was attacking both on the LNT front as well as on the point mutation front. So this guy was important. And when Muller gets his Nobel Prize, Muller highlights this doctoral dissertation by this person in his Nobel Prize speech, and that must have made that student feel particularly good. And I'm sure the student, if he was alive today, would not be feeling good about a paper that I wrote concerning, he and Muller and this dissertation. But in any case, what happens is that the study is actually weak, and if you really were to go into it, it ultimately is not going to stand up. But this was Muller's way out. So then what happens is World War II. And in World War II, as everybody knows, the United States built an atomic bomb and used the atomic bomb. And it was called the Manhattan Project. But part of the Manhattan Project that people really don't know about is that instead of just developing the bomb, the US also had a program that was to try to measure the effects or to study the effects of radionuclides that would come forth in the manufacture of the bomb. And they gave a series of big grants and contracts to the University of Rochester in New York State. And so when Muller came back to the United States, just at the beginning of the war in Europe, he got a job, believe it or not, in the town that I'm speaking at right now, Amherst, Massachusetts. And Muller's home is probably, I know exactly where it was. It's about almost 1 mile as the crow flies from where I am right now. And he had a little bit of walk across one of the main streets, and he would be right on the Amherst College campus. So you kind of can picture the old guy, walking back and forth to work. But while he's there, what happens is that Kurt Stern, who is a professor at Rochester, who came from Germany to Rochester. And actually, when Muller went and gave his talk with his Nobel Prize winning research in 1927, Kurt Stern was actually one of the co-directors of that big Congress. And I learned that that Muller asked Kurt Stern if Kurt Stern's wife could type could type out Muller's presentation, and she did. And so they had a relationship between each other, and Muller was well known. And so when he arrived there, Stern went to the United States Atomic Energy Commission, the AEC, and got permission to hire Muller as a paid consultant to the Manhattan Project. And so he was. And so and Muller and I got all the communication, all the written communication between Muller and Stern and all the people who worked for this for Stern on this Manhattan Project. I can tell you, Muller was an excellent consultant. Guy was detailed to the nth degree, and what he convinced Stern to do was to replicate, but in a much bigger way, what that graduate student did at the University of Edinburgh. It was very, very interesting. And so at this Manhattan Project, there were really two directions that the AEC had funded. One was a mouse model under this guy, Don Charles. And then the other was a fruit fly stuff with Kurt Stern. Now, nothing ever happened. This is another one of these nobody ever hears about, the mouse study. There were 400,000 mice that were purchased and used in the Charles research, okay? And there were no meaningful publications that came out of that research. Nothing that impacted at all the Manhattan Project, risk assessment, nothing. And from looking into all the personality traits and quirks of Dr. Charles, I came to learn that he was a very, very difficult man to please. He was extremely demanding, and he wouldn't publish something unless he was 300% sure that it was right. And he frustrated everyone because he held back on whatever they were working on. Ultimately, nothing was published. He eventually commits suicide in a Manhattan hotel. He does leave a suicide note that was read, but no meaningful results. People tried to publish a couple of summaries after he died, but it was too late and too insignificant and had no impact at all. And this was on the mammal model. That's what we really wanted the data on, was mice, not really fruit flies. But the only thing that was left standing was Stern's work, and that was going to be on fruit flies. So the Manhattan Project it's going to be advised in the United States is going to be based on what? Not mice, not a mammalian model, but on fruit flies, not what was designed. That's really an untold story, and it's really worth looking into. Well, as it turns out, Muller gets hired by Stern, and they try to do just what this student in Edinburgh did. Give a big dose and then have another group get the same dose and spread it all out. But they worked it into one PhD person would do this acute study, and then it would be followed the next year or so by somebody who would be doing the chronic one, where they could just spread it all out. This person who did the work for the acute one was a talented guy. His name was Warren Spencer, and so he did the acute study. Now, in this study, they looked at about 5 or 6 different high doses, and from high to low, but they were all acutely given. And as it turns out, they showed a linear dose response for this when they gave it all at once. It's exactly what Muller thought would happen, what he wanted to happen, the same with Stern. Now, and nobody ever criticized any of the work that the Warren Spencer report was about. I got the detailed letters of evaluation by Muller on this, and I have to tell you that when I got these letters, I made sure that I didn't read them until I had written my own detailed evaluations, because I wanted to compare myself with Muller, actually, in terms of how did I match up with Muller in terms of ability to criticize the study. And so what I found anyways were a lot of problems with this Spencer study. And these were problems that I'm listing here that Muller never listed. Now, this study had very poor temperature control, inconsistent instrument calibration. They would run exposures of animals on one day and not have a control group. Then one day they would have a control group and no animals. Then they would combine those groups. They took other groups where they actually gave the same total dose, but they gave them at a different dose rate. And that was very strange because that's really was the hypothesis they were trying to test. So, I mean, it was actually just counterproductive. Then they had many other limitations without going into all of them. It was actually a pretty poorly done study in many ways. And
let me ask about this. So this study overall was on the higher end in terms of the size of the dose. So they're giving large doses and seeing, and then they're basically counting up how many flies in the next generation look different than the parents' generation. And they use that to sort of say, hey, double the dose, double the different looking flies. Is it something roughly like that?
Yeah, it is, except the dose range, which they gave it all at one time, went from what I call relatively low, so to speak, to quite high. So there was a dose response range. And so there was still a high dose. Don't get me wrong, their lowest dose was still high, but it was far lower than had been done previously.
It wasn't 95 million times background.
No, that's right. Now, as it turns out in the next study, the attempt to calibrate this and compare it, this person took the lowest dose that was used in that Warren Spencer study, and he took that dose and he spread it out over 21 days. And so what that meant was they were giving the same total dose, but they were giving it at a dose rate that was 1/13,000th of essentially what the other one got all at one time. And so Muller would have predicted that you got mutations at all these doses when you gave it acutely. And so Muller predicted that, well, when you just take that same total dose, but you just spread it out in little dinky doses over the course of 21 days, whatever the lifespan was here, all these little doses would add up and be equal to the amount of damage you got when you gave it all at one time. That's his total dose versus dose rate hypothesis. That's what he claimed that this guy, the Indian graduate student in Edinburgh, showed. Well, Kasperi didn't find that. Kasperi found no damage. He found no increase in mutations when he did it that way. So when he got it tiny doses, spread it all out, nothing happened. And so he goes in and he shows it to Kurt Stern. Is exactly what Kurt Stern did not want to see. He wanted
just to be totally clear here, when you say nothing happened, you mean like no mutations happened? It was as if they hadn't been exposed at all?
That's right. It did not differ from the control group. That's right. It was okay. Similar to the unexposed control group, so that there was no treatment related effect. So that they could not claim that, these things accumulated and then they caused a similar amount of mutations. That just did not happen. So this was so he goes in and he tells his boss, Kurt Stern, we have a problem here. The problem is I can't support the linear dose response model. And so it's a serious challenge to this LNT. So guess what Stern does, of course? Stern says to Kasperi, I don't believe it. I'm not going to accept your data. And you have a problem with your study. And so he tells Kasperi, he said, the problem with your study and the reason why it's not valid is that your control group was highly variable and was its own mutation rates were higher than expected. And that's why we couldn't see something going on at a low dose. We couldn't see the linear dose response. It was really masking it. And he said, so I don't accept at all what you're saying. And so a wasted year. And so wasted 2 years. And so, but Kasperi said, well, wait a minute. And he goes into the literature and he finds a number of studies. And guess what? Those studies show, using the same model and so forth, they show that Kasperi's controls were right exactly in the middle where all the other data were. And that Stern's interpretation was actually wrong. And I actually knew that Stern was aware of these other studies because I I have even documented in letters him discussing these 3 and 4 and 5, 6 years before. So maybe he forgot. Who knows? He had selective memory, but I know that he was aware of some of these studies. Be that as it may, Stern actually did the right thing. He backed down and says, well, I guess you got me on that one. So he could not actually dismiss or discount the Casualty findings. So you might think, well, everything is good. That's the way science is supposed to be. Well, let me tell you what happens next, because Stern, he's really part of the Muller LNT group, and he wants to preserve this LNT concept. So what he does in this, in the write-up of the Casualty findings, and this is very strange. Now, the Casualty paper and the experiment was done far better than the Spencer did. And not that because Casualty was a better researcher than Warren Spencer, it was because he did it after Spencer. He saw all the problems that Spencer had, with calibrating the piece of equipment, with the temperature control and other factors, and all those wrinkles that were there for that first study, they basically cleaned it out. It's kind of like a football team. You always improve most from the first game to the second. And Casualty improved a lot in his methods and everything else, just because took advantage of what they learned with Spencer. But all those mistakes with Spencer, they compromised the Spencer findings. Casualty, they were kind of forced out, and his study was I consider it clean, and so what happened was, and his data were much more reliable. And so, however, when you take a look at what Stern did, and maybe Casualty was all part of this thing, but when in this paper I mentioned to you earlier that you have a discussion of your data. And then when they discussed the data that did not show a treatment effect, but showed a threshold, something that did not differ from the control group, Stern said, what to the audience, you might say, well, our data differ from that Warren Spencer acute study. We don't know why they differ. So do not accept our data or use it until it can be determined why our response was not linear like the Spencer is. And therefore, look at our data, but don't use it. He didn't put any constraints on the linear study, and that one was not done nearly as well. Now, I'd have to tell you, in part of the story, when you have what's the biggest and best study that's ever been done to date, and that was really Casualty, and you send it out, you should be very excited about your findings, but not so. They basically try to dampen it, poo-poo it, make sure it wasn't there, but to actually say that means that you think that there's a problem with your study. Now, and so if I were to, have the best study that I ever could possibly do, very, very unique, and I sent it out to a journal, and I told them, here's my data, but don't use it, anybody. Tell everybody who's reading your journal not to use my data. Well, they would just throw it back in my face. So how did Casualty's paper get published? Well, it got published because guess what? It was submitted to the journal that Kurt Stern was the editor-in-chief of. Now picture that, right? A little inbreeding here as well. And so it was submitted for publication on November 25th of 1947, and it was published in January of the next year. I'm quite sure it didn't also, neither one of these went out for peer review. It was just too short a time with snail mail and trying to get things ready for a January publication. You have to have that really well into the group by December. It was very interesting. Now, to me, what was important was that I knew, because I followed Muller's career, I knew that when Muller gave his Nobel Prize lecture, which was December 12th of 1946, that he stated unequivocally in that Nobel Prize lecture that the threshold dose response model, which had ruled the roost up until the present time, was no longer valid, that you couldn't support it, and it had to be, essentially put into the trash heap of history, and it had to be replaced by a linear dose response relationship. So I knew that was what Muller conveyed at the Nobel Prize lecture. But I'm saying, well, my goodness, if he had seen Casualty's findings, then he certainly would have known that Casualty's paper supported a threshold. he couldn't have actually stood up and said that there's no possibility because ultimately it's the strongest study and it's opposite of what he's saying. I said, so maybe Mueller never saw the Caspary findings, even though he was a consultant on it. Sometimes they don't show the consultant everything. So I got all the correspondence between Mueller and Stern and everybody else, and I somehow got it electronically sent to me at this desk I'm currently sitting at. And one evening I looked through all these, and what do I find? But lo and behold, on November 6, 1946, a letter from Stern to Mueller sending him the entire data set and the Caspary paper. And then the best part was on November 12th, Mueller sends a letter back to Caspary acknowledging the receipt of it, saying that he's looked it over, that he can't believe the findings, that it shows it challenges a linear dose response, that Caspary's a good researcher, I don't want anything bad about him, but you got to get some more money and you have to go and do this and see if it's right, he said, this is a serious problem. And so I knew that I had the smoking gun on that day. So as it turns out, picture yourself, you are Mueller, and you are, then now you know Caspary's work, right? And you're going to be going and giving the biggest lecture in your life, the Nobel Prize lecture. And you stand up in front of this audience, and what are you going to tell them? There's no possibility that there's a threshold when you've just seen the strongest data, and you can't criticize it. Well, that's exactly what he did. And so what would you have done? Well, if it were me and I was Mueller and I really believed in a linear dose response, but I knew that there was data contrary, and I was really part of that data, I was a consultant on it, I'd stand up and I'd say, you know, I don't know what the nature of the dose response in the low dose zone is, but I tend to think strongly that it's a linear dose response, but it's not resolved scientifically. More research has to be done, and hopefully it will be done. Is that what Mueller did? I think that's what an honest statement would have been if that was his position. But that's not what he did. Mueller attempted to use that Nobel Prize lecture and talks afterwards to actually give the impression that there was no possibility of a threshold, and you have to trash it when he was saying just the opposite in his written language. In my opinion, Mueller actually was devious and deceptive and lied at his Nobel Prize speech. It was really a very disappointing at best performance by Mueller, and nobody else would know because it was hidden. Now, Mueller, Stern would have known, Caspary would have known, Spencer probably would have known, and they all kept their mouth shut. They all kept their mouth shut in this conspiracy of silence because they knew. So Mueller receives enormous publicity, influenced regulators, the media, scientific community on public health, that the ionizing radiation at low doses is very harmful, very scary. And there's no safe dose. This is what he did. This is the man. Now you have to go back to who Mueller was and think back to this guy who basically didn't play by the rules when he tried to get his Nobel Prize study published. He won't show the data. He tries to beat Caspary. He basically is a person who has a series of character flaws. It's a series of character flaws, and basically they show up in critical times. They show up in critical times, and I didn't emphasize it here, but I actually have demonstrable evidence, that he misrepresented the scientific literature on the PhD students' work at Edinburgh, and with information shared with him between both student and him as well. I mean, Mueller, to me, and there'll be other circumstances that we'll just look at now, but here you can see that prior to his lecture, then Mueller knew of the supportive study. It was the strongest study to date. And interestingly, after his Nobel Prize on January 14th, 1947, he actually wrote a letter to Stern with his views on detailed views on Caspary's study. I have it, and he found no technical issues with the paper. None. 5 weeks before, 5 weeks after, he does not report any problems with that study. Now, following the internal review by Mueller and this Caspary and Stern paper. What they did was they removed the conclusion that there was a threshold, and they added Muller's name to the acknowledgement. Those were the only two things in a 40-page paper, 50-page paper. And so Muller got his perspective that there was no conclusion, and Muller's name was added as an acknowledgement. But Muller, in the aftermath, Muller had told them, we need those studies, that study replicated. So, but after the war was over, Caspary took off to be a professor, and the same for Warren Spencer. And Kurt Stern brought on a young master's student by the name of Delta Uphoff. And she was directed by Kurt Stern. And in the first study that Delta did to replicate the Caspary work, her control group was terribly low. And when she and Kurt Stern wrote their paper up, and they gave it to the Atomic Energy Commission, that was ultimately put in, was classified, nobody else could read it. Eventually I got the copy of it. And it said in the conclusion that because of the extremely low control group values, that their data were uninterpretable.
I'm sorry, low control group values of low rate of changes, small control group.
Right.
What?
It was, they looked at the mutation rate in the control group, because there's a background level of genetic change in all cells. And you're going to,
and they concluded that her control group values of mutation rate were unusually low,
that's
or
exactly correct.
is that it?
Exactly correct. Such that you couldn't validly compare it with a treatment group.
Because just on the basis of essentially saying that her control group was maybe faked, or I mean, how would you say that you couldn't trust it, given that it's low? What would be the basis?
It was outside what was reported in multiple published studies, way outside, and so it made them question its reliability.
Okay.
It made them very, and actually to take it one step further, look at the bottom part of this slide. And this is something I've never really seen before, but Stern blamed the low controls of Uphoff's replication on quote unquote investigator bias. It may have occurred because of it. So what does that mean? I said to myself, he's claiming in this paper that his name is on it, is that this low control group may have come about due to investigator bias. Well, whose bias? His bias, Delta's bias, the whole team's bias. Did they want to have to, because they were concerned with Caspary's initial thing that they could exclude it if they claimed that it was high, but if they had a really low control, then they could almost assure themselves that you would have a linear dose response. So there was, and so they basically acknowledged to the world that he was concerned that they got this uninterpretable findings because, well, I think he threw Delta under the bus because of I don't think he was going to throw himself under the bus. But that was written there, but nobody else could read it. I got it to read it. So this is actually pretty amazing for me to see this. So as it turns out, Delta's second experiment had the same very low control and was also viewed as uninterpretable. She does a third study. This time the control group came out okay, but the low dose response was like 3 or 4 times higher than you could have ever expected, even if it was a linear dose response. And Stern just threw his hands up in the air as like there was nothing that he could salvage from any of this that was going on. And when you are coping with the fact that Dr. Charles wasn't doing anything that was going to be publishable, the whole Manhattan Project up at Rochester was actually not going to be very productive at all. And so, but Stern basically pulls a rabbit out of the hat. And 6 months later, he decides to take these 5 experiments and put them into what he calls a meta-analysis. And that is, he takes the Warren Spencer study and the Ernst Caspary experiment and the 3 experiments by Delta Uphoff and puts them all together. And he basically presents them in a 1-page paper, a technical note in the journal Science. And what he does is he discounts the Caspary study all over again. It's like it was back another 1.5 years ago. Discounts it again. The Delta studies, which were uninterpretable, guess what? He now finds them interpretable, but he never tells the readership or anyone that I know of that he 6 months before, 9 months before, he and Delta in writing in their report to the federal granting agency calls her data uninterpretable and on the basis of investigator bias. He hides that entirely, not shown. I'm the only one who finds that. So he publishes this in the Journal of Science, and it's in the Journal of Science, and it's showing a linear dose response relationship, and this becomes the key publication. It's going to be the basis for the acceptance of a linear dose response by the US government. This paper right here. This is actually pretty amazing. And so Casperi's study, which was marginalized, I don't know, I don't know how it actually happened, but one can imagine. I found a series of papers after this fact in which people said, oh, Casperi initially found no changes, but the work had to be discredited because it had a high control group and so forth. And we know that was actually not shown to be true, but rumors came to be. People wrote about this, and Casperi never actually challenged that in the literature. Now it's interesting along the same line. During this time period of the debate over Delta and so forth, Mueller is in big time debates with his old nemesis, and that's Statler, Louis Statler at Missouri, over control group changes and mutation and so forth. And Mueller has hundreds of thousands of reproductive information on his control group and their mutation rates. And I have letters from Stern to Mueller asking him to share his data on control group mutation in all his work at Amherst College. And in these letters, this is actually pretty amazing. They go and they ask him, well, which control group is supported, the Delta control group or the Casperi? And he says unequivocally, Casperi's matches up with mine, not Delta's. It's actually pretty amazing. It's all in the literature. And then when he leaves Amherst College and he goes to the University of Indiana, his work there, it still matches up with Casperi's, not with Delta's. It's all in the literature. And yet, I have to tell you this tells you something about Mueller. In the early 1950s, Mueller repeatedly and inexplicably to me challenged the Casperi findings in writing and saying that his control group values were aberrantly high. And it was and yet the data of Mueller, both before and after the Casperi paper, they fully support the Casperi interpretation. So why would Mueller make such knowingly false comments and do so repeatedly? And well, I mean, Casperi or Delta or Stern or others could have just discredited him. But nobody came forward. And I think that they were afraid of the old man anyways. I mean, he's a tough guy to deal with, and he could cut you down. And so, but they all supported LNT. And Mueller, if they ever came forward, they would have also exposed that Mueller had lied during his Nobel Prize speech. It was really circling the wagons and not saying anything. And again, a conspiracy of silence here by people who are in the know. If you really want to look at ethics in science, you can look at this as a good example of a lack of ethics in science. So Stern goes forward, publishes some highly acclaimed genetics textbook, multiple editions from 1950 on. In that textbook, he uses the data of Uphoff and Spencer to provide the linearity interpretation, and he totally ignores the Casperi findings. Now, all this is going to converge on a big event that happens in 1956. And this is in the aftermath of lots of Cold War activities between the US and the Soviet Union. So a lot of atmospheric testing of atomic bombs. There's concerns with fallout. And what happens in 1955 is that the Rockefeller Foundation, they basically go to the National Academy and they give them a chunk of money, and they want the National Academy to get 6 committees together to look at the environmental, public health, medical implications of ionizing radiation and radionuclides. And now this is very significant. Up until this point, there have been different kind of medical committees and so forth on radiation over decades, actually. And when it came to medicine, there was a medical committee, and there were always geneticists that were on the medical committee, but the committee may be made up of 10 to 12 people. There's always a geneticist or two on the panel, amongst other kinds of specialties. Because of that, the principal areas of medicine, they tended to support a threshold interpretation for radiation. The geneticists tended to support Muller's position, which was, no, that's wrong. It's a linear dose response. So the geneticists were always frustrated when they were on medical committees because they were always outvoted like, 10 to 1, 9 to 2, whatever it was. However, the radiation, the genetic.
Well, let me ask you about that. That's very interesting that there'd be such a difference in views between the two communities. The medical community that believed there was a threshold, was this on the basis of studies and data? Was it based on hypotheses about the underlying processes and why there should be a threshold? Why was there a difference there?
I think it's a couple of things. One is that the world of pharmacology that really led by the British back in the early 1900s, had, and by the late 19-teens, had made basically a judgment that the basic dose response in biological systems was a threshold, and even for highly toxic chemicals, and and for many pharmaceutical agents as well. So there was some general medical belief in the reality of thresholds, and I believe that they saw radiation is not particularly unique. They didn't actually at that point know or understand would a mutation be different in its response than all the other kinds of effects that are measured. And so but I think that they were initially impacted because they all come out of, pharmacology was like a queen of the sciences, in medical school. Radiation would not be one of the queen or kings of the sciences at that particular point. And pharmacology still is. It's just a gigantically important discipline. And the other aspect of this is that from the point of view of trying to use like radium as a medical treatment for tumors and things of that nature, and how the radiation would be held, or the use of X-rays in the treatment of various types of conditions. I believe that the medical community was probably in their self-interest trying to steer away from a linearity interpretation because it may scare patients, and they didn't want to do that. So I think there was some self-interest involved in how they looked at it, but there was also the historical aspect that came in from the pharmacology history, and they were all trained in pharmacology at some point. They all took courses in it. And none of them really had courses in radiation. They all had courses in pharmacology, so they had a view that permeated them.
Okay, so on the pharmacology, the threshold issue on the pharmacology, is this a threshold in terms of useful medicines have no effect before a certain level, or also from the standpoint that if you go to too high a level, it's no longer beneficial but actually dangerous? A bit of both. Could you say more about why there was a threshold model that showed up in looking at pharmaceuticals?
Actually, it's typically for pharmaceuticals, it'd be a threshold for when you begin to show the effectiveness of an agent. So at this level, the aspirin starts to work, that sort of thing. And so below that, that's why you might need 2 aspirins instead of 1/2 of an aspirin. So they'll help you on getting the right dose. Okay. But also when it comes to toxins, the threshold would be probably equally applicable there too. So and when it came to radiation, how they saw this is that if they were to and this is interesting for people who were exposing women who had breast cancer back in the day when Madame Curie was alive, and she provided little ampules of radium. They would the technician. It sounds so crude, and it is, and it's dangerous, that they would actually hold with a little tweezers for a period of time the radium over the breast area or where the tumor was that they were trying to treat. And the some of the technical people I know at Columbia and otherwise, they were concerned with their technicians being exposed and to the radiation, and they did not want to not treat the patients, and so they came up with this view that it was safe to hold the radium until your skin on your fingers showed a color change, became red. And that was a sign that a biological change would happen. And it only happened after a certain time period of exposure. And that was one of the bases, and you can see how crude it was, for there being a threshold. It wasn't very good. But so that was coming in from the industrial hygiene, radiation hygiene sort of perspective, using some of these new radiation related, but still crude from our point of view. It's 100 years, it's 90 years old. It's 100 years old, almost, perspective as it's entering into treating of patients. So it was within that framework that concepts of threshold and what was acceptable practice and what they would how they would expose workers as well as patients, and things got a lot more quantitative, over time, but the ideas of how to cope with this was pretty rudimentary. So basically what happened is that during this 1920 to 1950 period, there was a growth in knowledge. But what happened was that Muller, after 1930, after his finding in 1927, he just became a strong advocate that there was no threshold, and that we could expect to get mutations, and that mutations were harmful to the next generation. He also made assertions that there would be risks of cancer, and he claimed that based upon the two studies that I just earlier shared with you, that the dose response was linear. Now, during this time period, I would have to tell you that people began to improve their studies, and they began to improve the quantification of exposure so that you could refine your X-ray dosing, and you could expose plants, or you could expose whatever organism you were working with. And it was interesting, in the late 1920s, it was 1928, but published in 1930, Statler actually published a 13 dose study with radiation and effects on corn. And he found that using lots of specimens, that he found that the lowest 3 doses essentially showed no treatment effect. And at higher doses, you saw adverse effects. So he himself saw a threshold. In his writings, he claimed that there was a threshold. There were a number of other studies like that were quantitative, and that used improved methods of analysis and detection, like Barbara McClintock was coming up with. And they actually supported a threshold. I'd have to say Muller never cited those papers in his work. He was remarkably blind, or filtered stuff out that didn't actually go along with this. Well, fast forward up here to 1956, and there's some serious problems in this country and around the world with trying to cope with international instability between in the Cold War, but also trying to figure out how dangerous is the radiation that we're being exposed to, and increasing. And so a committee was put together of, you might say, the best and the brightest, but and this is going to evolve in 6 areas. One area now, and this becomes very significant, having spent a certain amount of time talking about the medical versus the genetic perspective here. And that was that and this was the very first time in this country when the National Academy of Sciences appointed the committees, it for the very first time appointed a separate genetics panel. They had never had the freedom and the power ever before. They were always submerged within a genetics panel, in a medical panel. Now they had their own say. Now the interesting thing here is that back in those days, the federal government didn't fund academic research. It was really funded in this country and some other countries by the Rockefeller Foundation and the Carnegie Institute. But the biggest one was the Rockefeller Foundation. All the academic geneticists that were going to be on this committee had been funded for a long time by the Rockefeller Foundation. At the time that Mueller joins this committee, the University of Indiana had $4 million or so of $1 million back in the 1950s. It's a lot of money even today to study genetics. Even more money was given to, by far more money, out to California Tech in their biology department, and a lot of it funneled into radiation research. And now the director of research for the Rockefeller Foundation, he was put in charge. His name is Warren Weaver. He was put in charge of this BEAR, which stands for the Biological Effects of Atomic Radiation Genetics Panel. So he's a mathematician, but he's put in charge of this. But he's the research director for the Rockefeller Foundation, and he has given grants to all these people for many, many years, going back actually even 20 years before. He knew Mueller extremely well. He knew them all extremely. He knew their beliefs on the dose response. Okay, he knew their beliefs. Now I have to tell you also that the president of the Rockefeller Institute for Medical Research, which is today called Rockefeller University, and the president of the U.S. National Academy of Sciences, his name was Nat'l Bronk, and he held both positions. Hard to believe this was true. So a person on the Rockefeller Foundation board and president of the Rockefeller University is also the president of the National Academy of Sciences, all in the same. It's all inbreeding. So wow. When Rockefeller decided to give money to the National Academy, they were just giving it to their own person. And they were funding everybody else in the genetics world, and they chose everybody, essentially, they knew their views. So it's kind of like they, essentially choosing a jury, and the views of the jurists. Now, they all came in, and I knew at the time that the threshold dose response was the one that was prominent. It was the publicly accepted model, the one used by the US government and other governments around the world. But I knew, because I knew the history, that by the time this BEAR committee was over, they decided to reject reject the threshold and accept LNT, just like Mueller had said 10 years earlier at his Nobel Prize lecture. They just essentially just took Mueller's talk and made that their policy. But I wanted to know, I said, well, did they argue about this? How did they debate this? Was it really a big brouhaha? How did the threshold model go down fighting? So I got all the transcripts, right? And I looked at it, and I was wondering, well, who supported the threshold? Who supported LNT? Who was the biggest, most articulate spokesperson? And I got an absolute shock when I read this thing. Within very early on in the panel deliberations, Tracy Sornabon from the University of Indiana, who's on the committee, he goes up and he reads the radiation mantra. It's like the Apostles' Creed. He tells them this is what the basic beliefs are of radiation geneticists. It was Mueller's beliefs. It was every exposure has cumulative damage. There is no repair. All damage is irreversible, and the dose response is, guess what? Linear down to a single ionization. He read that into the record. There was no debate. It was like it was all over before it began. Threshold was out, LNT was in, and so I'm over there like a reader. I'm waiting for the big debate, and there was no debate. It was just a takeover. It was just a takeover. If you had an honest panel that was put together, then you'd have half the people were on one side and half on the other, or something like. You'd have had a debate. There was no debate. I can tell you that. It was very disappointing for me to see that. Now, the interesting thing on this is that You might think that was the best thing that ever happened to LNT. It was the worst thing that ever happened to LNT. Why? Because in the course of putting this committee together, they had enough money for about 6 or 7 or 8 meetings. So they had money in the budget, and they forced these guys to come back to meet so many times. And there was nothing really to do. They had decided to go linear right from the start. So Warren Spencer, bureaucrat that he was and all that sort of thing, he decided to give these very brainy people make work sorts of things, right? And so the make work project that he gives them is going to do them in. And that is that he tells these, now on this committee, there are 17 people and 13 of them are geneticists, okay? And he gives the geneticists the challenge. And the challenge is that let's assume that every adult in the United States gets their gonads exposed to a certain level of radiation. I want you to come up with an estimate of how many kids will be born with birth defects in the next generation and out for 10 generations and things of that nature. Gave them three different challenges. And so he said, I want you to write this all down, send it to me in a month, and we'll discuss it when we meet again in New York City. And so what happens is that he said, this is good because you're all different. There's somebody here who's a bacterial geneticist, a paramecian geneticist, a Drosophila geneticist, a clinical geneticist, a population geneticist. They're all mouse geneticists. There are all kinds of geneticists on this committee. He says, this is good. You all have different viewpoints. And I'm going to force you to all believe in LNT. So you'll have to force you to have LNT. But then I want to see our perspectives become much stronger if we have a great confluence of views. But he was all steering them into LNT anyway, so they should all kind of agree. Well, the interesting thing that happened here is that when the results came in, guess what? They didn't agree with each other. They profoundly disagreed with each other. They were miles apart. And when one of the people on the committee who was collating everything saw this, it freaked him out. And he writes a letter to Weaver, the chair, saying, hey, if people are going to see how much we disagree, then nobody will ever accept any recommendations come from us, because if these guys can't agree, then why should we follow anything that this group sends to us? And so this was a really critical moment in this committee, and it's the first of a couple of critical moments. I'll just complete this loop to show it to you. So what do you do in that circumstance? If everybody's agreed on LNT, that was perfect, that was great, but when they apply it to try to estimate damage, they can't come up with anything that's close to similar. They're all over the lot. So what the guy, his name is James Crowe, he's a university professor at the University of Wisconsin. And what Crowe does on his own, at least, right now, although I believe he consulted with Mueller on this, and I have reasons for that, he removes the 3 lowest estimates without getting approval. And so now what happened is that there were 13 geneticists who were invited to do this. One geneticist quit the committee because he had too many academic obligations, a guy from Baylor University. We were left with 12. Of the 12, 3 decided that this was a crazy assignment and we're not going to do it. It was just too much uncertainty. The other 9 actually accepted the assignment and all provided detailed write-ups. And I have copies of all 9, by the way. I have them right behind me in this office. Copies from all 9 of what their estimates were. Now, and these estimates are the ones that differ from all over the place. And essentially, Crowe removes the lowest 3. Now basically, how I discovered this was that when the committee had finished all its deliberations, it wrote up its report and published it in a very prestigious journal called Science that we talked about. So when I was reading the Science article by this Bayer 1 genetics panel, they told the story that I just told you. They said, well, we challenged the committee. We wanted them to estimate the number of mutations, of birth defects that would happen in the first generation and so forth. And if they got a certain level of radiation. And so when we challenged our panel of 12 members, 6 agreed to do the review. And this is what they said. And I said, wait a minute, He's saying they challenged 12, but 6 agreed. I said, I have 9 reviews sitting behind me. So what the heck happened to the other 3? So I had to go and figure out which 3 had been removed. And it was not too difficult to figure out which 3 were removed. And they were removed. And what that did was it reduced the uncertainty down to about 750-fold from many thousand, down to 750-fold. It was still considered too big, apparently, by this committee. And so without coming up with any rationale for anything else, they then, when they published the paper, they said, and the variation was approximately 100-fold amongst the panelists.
Was there an objection raised, or what about the 3 whose studies were dropped? What did they say?
Well, it's interesting that you raised it. I've thought about this a lot and written about it, actually. And you, I mean, if it were me, I'd have got so upset, I'd have banged my hand on the desk. I'd have written a minority report. I'd have done something. But none of the 3 did. Now, why didn't they? And I have reasons that I'll get into for that in my next thing. So but the point that I'm going to make right now is that in the world of science, you have to tell the truth. You can't falsify or fabricate information. You have to reveal the research record. You have to actually reveal it. You might disagree with it, argue it, say you don't agree, why you don't agree, but you really can't misrepresent the research record. When they wrote this article in Science that they all signed off on, they claimed that 6 adopted the challenge, and I know that it was 9, and I have the documentation for that. And they said it went down to plus or minus 10 for a 100-fold variation, and I have it down to 750 even from that. They actually committed, this National Academy of Sciences group actually committed scientific misconduct, all of them. And even if you disagreed with it, they all did it. And so, and this was totally undetected until I started digging into this. And basically, now I have to tell you that outside academics, they wrote and they requested the scientific basis for the linearity and so forth. And the panel refused to do it. It went up to Detlev Bronk, the president of the National Academy, and he refused to have the committee provide any written basis for how they came up with their LNT recommendations. Now, one thing I'm going to go back to here, and that is when this panel was first put together in November of 1955. That's essentially 10 years after the dropping of the atomic bomb. During those 10 years, the US government and the Japanese government put together a group called the Atomic Bomb Casualty Commission, and was overseen by the National Academy of Sciences from the United States. And they did a couple of studies, and they're still going on actually, even today. But one was to take a look at mutations in the offspring of people who were exposed to the atomic bomb. And a member of the Bayer panel was in charge of that big study, a University of Michigan professor by the name of Jim Neal. And it was a gigantic study. And by the time 1955 rolled around, they had been studying nearly 75,000 children for mutations. Now, you might think that kids who were born to parents who were exposed to the atomic bomb, well, they might be born with 3 eyes, 2 ears, 3 ears, all kinds of defects, what have you. Anything could happen, right? However, and they have thousands of people working on this study, trying to do it in the fairest, detailed, whatever fashion possible. And I'd have to tell you that they found no increase in mutation, none, after 10 years. Now, I also want to tell you that we can fast forward from 1955 to the last big study, which was 2015, and they still haven't found any. Okay, so with all the refined new advances in looking at gene changes, and they have still not found any significant damage in the offspring. Now, this over here is challenges exactly this whole concept of a linear dose response. These people were exposed to very high levels of radiation, and it didn't affect their offspring in terms of whatever sensitive indicators over time that happened. But up to the 10-year period, everything was negative. So Jim Nat'l shows up to the Bear Committee. It's hard to believe, but it's true. He gives them a 250 to 300 page report, 10-year report, and he wants them to evaluate this because it's perfectly germane to what this Bear Committee is about. They're looking at the effects of atomic radiation on human subjects. And what would happen if their offspring, if parents were exposed to ionizing radiation? Well, Jim Nat'l has done the study with the Japanese, and it supports what? It supports a threshold. And so guess what happens at this meeting? Mueller speaks up. He ridicules the study, he ridicules Nat'l, and gets everybody to not give the study that is a National Academy of Sciences study, a study that is involved thousands of people working on it between two countries, trying to do it in the best possible way, and they refused to evaluate it. And they decided to base their entire recommendations on what? On Delta Uphoff's and Spencer's fruit fly studies, which are highly compromised. You think I'm making this story up, right?
It does sound like it almost has to be made up. Let me just repeat that back one time to make sure I got this. So the National Academy of Sciences has pulled together this biological effects of atomic radiation study group or task force or whatever it's called. And in their attempt to assess the impacts on human beings of atomic radiation, they ignore the huge study on what happened in Japan.
Exactly right. You get you got an A. And I'd have to tell you this, that despite the fact that I've worked for a long time on this, I didn't know this. I discovered this about a year ago, a year and a half ago.
Wow.
It's somehow it's out of everybody's consciousness. Nobody ever, and I have published it, and it's out there. It's in people's faces to read. It is absolutely true. It's absolutely true. And now I have to tell you, it gets more interesting. So if you're Jim Nat'l and you've been working on this for 10 years, right? It's like if you've been doing your dissertation for 10 years and then nobody wants to look at it. They just flush it down the toilet, really. I mean, how angry was Nat'l, right? How angry would you have been? I'd been really angry. And then at the same time, you're not serving the American public or any public. You're not serving humanity. It's a waste of money, waste of time, waste of everything. And Mueller goes on and he bases things on fruit flies data that had problems with it instead of this human data. I'd like to just raise the ethical question here. Was that committee very ethical? Was Mueller ethical? Was the National Academy of Sciences that's overseeing this ethical? This is who guides the United States. I mean, these are our leaders, okay? Those are our leaders. So what would you do if you were Nat'l? Well, Nat'l was, you might say, a mite angry. So what did Nat'l do? He knew that there was a parallel committee over there in Britain. So what did he do? He slipped the report over to the Brits without his committee knowing it. So what do you think happened? Well, they looked at it and they had many questions. They wrote back and forth. I've got all their, as much as I can buy and purchase and so forth, all the communication between him. And what happened is that in the end, they highly praised his study in their report. It markedly affected their recommendations for mutation risk and so forth from radiation. And they're vastly different between these two countries. And it was because of Nat'l's findings in large part. And essentially, work done by this country, given to the Brits used it, Mueller and those people, and who are supposed to be serving this country, they just ignore it. Now, the story doesn't end there. It gets actually more interesting. You have to understand that Mueller he's a Nobel Prize winner 10 years out, and he can be a very tough customer. The smartest guy in the room could cut you down and wouldn't mind cutting you down either. He's got a reputation for that. And Neil, much younger. And how do you take on the big bear who's a Nobel Prize winner, right? Who's going to go after you and chop you off at the knees? But I'd have to say, give him some credit. There was that summer in Copenhagen, there was a big international radiation conference followed by a World Health Organization expert panel. And Mueller and Neil are going to be participants in both. And Neil opens up his presentation at the conference with an absolutely devastating, in your throat presentation directed towards Mueller, all about how you can't rely upon fruit flies for extrapolation to humans. The best study for man is man. We have all these data from the atomic bomb study, and they didn't show any effects. Made headlines in the New York Times, actually, and it developed into a battle between Mueller and Neil. And so the New York Times writer said, well, there were two groups. There were the malarians and the anti-malarians. If you want to take a look at the language of the New York Times back in the day. And so what happened was that it spilled over to the World Health Organization meeting, and there were all kinds of write-ups that went on there. Mueller tried to prevent Neil from being able to make contributions to the World Health Organization, to the technical report. It led to geneticists choosing sides. Who do you love more, your mother or your father, right? Some supported Mueller and some supported Neil. When Mueller tried to get the chair of the committee to not permit Neil to speak, a number of the British geneticists threatened to walk out of the meeting. They calmed things down, but when it was all said and done, what happened was that Neil and Mueller tried to resolve their differences like adults back in this country, and they went to Oak Ridge National Labs, and a few months later, with the head of the Oak Ridge program trying to meet, who was also part of the Bear Committee, trying to mediate their differences, actually. And these guys, they just disagreed. But at the same time, Mueller tried to hurt the reputation of and threaten the future career of a much younger Jim Neal. And Mueller was older. He was the Nobel Prize guy, and it could be a nasty guy. And I have all the threats in writing. I have all these letters back and forth. It's in the papers that I wrote. And so what happened is that Neil decided that he fought Mueller, and then at the same time he tried to. He was what's the right word, confused as to how much to break with the group, not to break with the group. He made his point, and then he backed down, and kind of I would have to say let Mueller rule the roost, but the damage was done. It was his study, even though they wouldn't look at it, his study had made it out into the New York Times and elsewhere. And people knew about it. And it actually hurt the credibility of the Bear Committee, because after they were saying there's no level of radiation that's safe, here's this biggest study with humans in this most terrible situation of two atomic bombs, and didn't show any damage. It showed that this whose study was right, Kasperi or Stern or Spence's, right? Yeah, it was Kasperi's that was more consistent with these data. So basically, are you going to ask a question?
Oh, no, go ahead. I was just, I'm just, this is pretty astonishing.
Well, and so what happens is that while Mueller and Neil are really duking it out over in Copenhagen, the Warren Spencer decides that that he's going to resign from being the chair of the Bear Committee, because he actually had to go back to Rockefeller, and he was trying to develop a research program for these geneticists, and he had to figure out how to get them the money that were more or less promised for the kinds of recommendations that they came forth with. And the person that they put in charge of the new committee was from Caltech, a future Nobel Prize winner by the name of George Beadle. Now this is it gets very interesting here, and so, but I will return to this in a second. Okay, I just want to let you know that within a year or so after this recommendation, within 2 years really, one of the committee members, his name is Bill or William Russell. He was at Oak Ridge National Labs, and he was present actually when Mueller and Neil had their little tete-a-tete down in Oak Ridge. But in his laboratory, he did something that was discovered something that was basically unique, because Mueller was saying that remember that the old mantra that all damage is basically bad. It's irreversible. It's cumulative, and ultimately can't be repaired, and ultimately it's a linear dose response. Well, working in the laboratory at Oak Ridge, Bill Russell, he's working with cells called spermatogonia and oocytes. These are the reproductive cells for male and female organisms, and he's working with mice now, no longer working with fruit flies. And what he finds is something really interesting with these cells. He finds that when you give a dose of radiation to the male and female, that all the dose isn't actually the damage isn't cumulative. He's with the female, he finds that you can keep giving higher and higher and higher dose, and the female has the capacity to repair all the damage that you give it, until you get somewhere, I think he estimated around 27,000 times, I think it was 27,000 times higher than background. And the male wasn't as good, but it still had that same capacity. And so he was able to show that in the female, for sure, that basically there was a threshold, and you had to go up pretty high on the scale before you wouldn't see a threshold. And so Neil published that paper in the journal Science, and it got a major publicity. And so this was a very significant. However, the female showed a clear threshold. The male was able to reduce about 70% of the damage, but couldn't get it all done. Now, this work went from 1958 and pushes all the way through the 60s. And then in 1970, see if I can get this. Yeah, 1972, a new panel is put together, and it's called the biological effects of ionizing radiation panel. And they were going to look at this Russell work that I've just mentioned to you here, that was published first in 1958. The significance of this work is that it actually suggested that there was such a thing as DNA repair. And that was ultimately proven to be true a couple years later. But it suggested the existence of an adaptive response. It suggested that there was even the possibility of thresholds. In fact, in the female, it was really shown. So now what happens next is what do you do with this information? So Mueller dies in 67, but in 1970, when EPA is created, they put together this Beer Committee, the biological effects of ionizing radiation, and they say, tell us what to do. And so what happened is that this is what they did. They looked at the data from Russell, and they said that Bear Committee in 56 had made a mistake. That the Bear Committee had said that there was no possibility of repair. But they said, yeah, repair does exist, and we made a mistake. And the reason why they made a mistake is they based it upon mature spermatozoa, which are the final stage of sperm before they basically enter the reproductive interaction, with the female. But up until that point, the younger sperm cells, they have a full complement of DNA repair. And so what Muller was working with was the final stage mature spermatozoa. He was looking at the most the group that was at greatest risk, and so it was really a very different sort of a cell. But in this situation here, Russell had really shown that there was such a thing as a threshold. But the Bayer Committee, in advising EPA what to do, they said, well, even though there is a threshold, and we see it in females, we haven't seen it yet in the male. So we're going to, even though they were wrong and there is such a thing as repair, we're still going to accept the LNT as our mode of action. And in fact, in 1975, the EPA accepts this Bayer 1972 interpretation. And this is because the Russell studies were based upon probably 1.5 to 2 million mice. It was gigantic studies. And this became like the stable reading that could offer some credibility to an EPA recommendation. So it's all based upon this, but they actually had the basis for a threshold here if they had wanted to. Now I want to take you to 1996.
Let me ask one question about what we had just gone over. So you talked about the difference between the sperm cells and the oocytes. Were they were specifically focusing on these cells in the mice to get a sense of, because they wanted to look at germ cells and the impact on future generations? Is that why they focused on these cells, or did they look at other tissue as well?
They looked at other other stages of development, but the results were coming back too inconsistent. And they had much greater consistency in their findings when they looked at what I'd say more. They're not really quite germ cells, but they're much more closer to a germ cell than an advanced reproductive cell like the mature spermatozoa. But they also thought that these were more valid because to look at, and even the general committee did, because in a person's body, 75% of the time, the reproductive cell that's going to be present is going to be the spermatogonia, and it's only for a very short period of time that these other stages are present. And so there's going to be a mixture of cells that are present, but basically it's strongly dominated by the presence of the oocytes and the spermatogonia. So in essence, this was considered to be, in the end, probably the most proper cell to look at. But in the end, it was also found to be cells that had very strong capacity for DNA repair. And this was really very, very interesting finding. And the other thing, from a human point of view, the reason why losing the DNA repair in the male isn't of significant value, because it's obviously happened in the course of evolution. So, but the reason, I don't know why this was lost during the course of evolution, but when that sperm fertilizes the egg, the egg brings with it her full complement of DNA repair and cleans out any residual damage that might be present in the male. And so therefore the species is preserved. And so it's a very interesting evolutionary trade-offs that actually happen. But in effect, what happened here is it was because of seeing that the male did not have as good a performance in the Bill Russell study as compared to the female, even though there was strong evidence of repair, that they still held to an LNT interpretation. Now, fast forward to 1996, and this fellow who's still alive, his name is Paul Selby. He has worked for many years there. He's a PhD in genetic toxicology, worked with Russell, and Russell was his mentor and so forth, and knew the situation extremely well. And he had to go back into the old archives of all the data and dig certain things out. But while he was doing that, he noticed some irregularities with the Russell control group data going way back into the 70s, and it bothered him, and he didn't quite understand it. So he went back night after night, and he began to sift through from data and findings, and he came to the conclusion that there was something seriously wrong with the control group that had been used by Russell in all these big mouse mutation studies. And in fact, he was a very quantitative man himself, and he concluded that in fact the mutation rate that the Russells used was in fact wrong by 3 or 4-fold, way off. And he had also suspicions that this might have been deliberately done. And so he put together a large dossier of information. It's all secretly done. And he then shared that information with people high up in now what's called the Department of Energy. And when they saw what he had, they of course didn't say you're right, but they said you have standing and you have credibility such that this needs to be looked at. So what they did was they put together a committee of 4 people, top level people who didn't have an axe to grind, so to speak, in this debate. And they brought in Russell and his wife and others, and Selby they all had to testify, so to speak, and debate each other. And I have the transcripts and everything I got these under Freedom of Information and so forth and so on. And in it, basically what happens is that the panel doesn't really want to come out and say, except Selby's assertion that there was something that was mischievous, put it that way, a deliberate deception. But they came out and supported him by saying the Russells had made a significant error, and they wanted the error cleaned up in the literature. And so they requested the Russells and Selby to write their own papers and to publish them in the literature. This is very highly technical stuff, okay? They publish it in the leading journals. Russell's published theirs in the Proceedings of the National Academy of Sciences, and Selby publishes 2 papers in a genetics journal. And even though they disagreed with the extent of the error, but just telling you how much the Russells thought they were in error. The Russells thought they were in error. They agreed that they were in error by at least 120%. Selby had them at larger error. Now, that's a gigantic error for a control group. You're off by 100%. No, you're off by 120%. It's gigantic. And so they wrote this up, very technical in the literature. I don't know how many people, 25 years after, really got into it. Whoever wrote follow-up letters and so forth, or articles, Selby seemed to have won the argument as far as I was concerned. However, what I did was I went back in, I just took the Russell correction because that was the simplest one, and I wasn't, I didn't have any great axe to grind. But what would happen if you used the correct control group, assuming Russell was right, and you went back to 1972? I'm not even using Selby's. So when I went back and did it, what I found was that if you applied it then to the data, the female would have showed that low doses of radiation were protective, and it would have showed an hormetic dose response. The male would have showed that there was a threshold. In other words, EPA rested their judgment on the Russell findings that were based upon an incorrect control group. If the correct control group data had been used and the EPA followed the data, then the data would have indicated that they would have adopted either a threshold dose response or a hormetic dose response or some combination. It would not have supported a linear dose response. So I published that in a series of papers. I got comments from Selby and so forth and others, and looked at it, and it's out there in the literature right now. So the whole basis of this National Academy recommendation was based upon a mistake, acknowledged mistake by the people that they based it upon, and EPA still doesn't change anything, actually. And this doesn't even deal with the whole issue of them refusing to look at the James Neal stuff. Is that actually pretty amazing? And so, but the story, if you think the story is bad up until the present time, it actually continues. It continues on this same path. So there's, and this brings us to our, you might say the final chapter in this story, and it's the life of Edward B. Lewis, another Nobel Prize winner, 1995, I think he got it, and in genetics. And this is about Ed Lewis and his work on leukemia and LNT. And this is how one paper and one person really actually could make a difference. And so who was Ed Lewis? Ed Lewis is a future Nobel Prize laureate, 1995. In 1997, when he was young, a young Cal Tech genetics professor, he was a fruit fly guy, a fruit fly expert. Now, he doesn't have any education in radiation, cancer biology, leukemia, epidemiology, risk assessment, and biostatistical modeling at low dose. He doesn't know the field, actually. So how does he get involved with radiation, leukemia, and risk assessment? And how does he change the whole world, actually, to look at this? Well, how it all happens is he's at Caltech. And at Caltech, it's back in the 50s, it's a real center of environmental activism. And you had some pretty big names who were very active. The most prominent would have been Linus Pauling, who wins the Nobel Prize in 1958, who basically also wins a Peace Prize in 1962 for trying to force the US and the Soviet Union to sign a nuclear test ban treaty. And so he's very, very much in the public's view. And then you have two other big time geneticists who are also very active as well, Alfred Sturtevant and George Beadle, and these have unique relationships with Ed Lewis. Sturtevant was his advisor when he got his PhD at Caltech, and George Lewis actually was employed his wife, and so they knew he had a unique relationship with these people. Now, but in 1955, July of 55, Beadle sends a memo to all his biology faculty trying to encourage them to assess the impact of fallout on people. Now, whatever it was, the only one of the faculty who decides to take up the challenge is none other than Ed Lewis. And this actually leads to what will be this major paper that changes the way we think about radiation and leukemia and then other cancers. It all happens in 1957, very close in time to this Baer Committee. Now I have to go back to this Neal paper, because the Neal paper that they refused to give standing to, that Mueller spoke up on and so forth, and that created the big brouhaha over in Copenhagen just when George Beadle was becoming the chair of this committee. So just picture this: you becoming the chair of this Baer Committee, and the committee is splintering and breaking up because of the big fight between Jim Neal and Herman Mueller, and people are taking sides. It's like the family feud, and so he's going to try to pull all these people together, but there's no letting, basically the horse has already escaped from the barn. That's Neal's report is now out. There's no way to go back and try to sugarcoat this. And that is that Neal's paper discredited the whole recommendation that came out of the Baer Committee, just discredited it. And so it's interesting. And so what does Beadle do? Beadle then he tries to redirect the work of his group, not to genetics for the sake of birth defects, but genetic damage, mutation for the sake of cancer. He changes the focus. And so what happened was that and he tries to get Lewis to actually redirect himself along those lines. Now, Lewis doesn't know anything about leukemia. He doesn't know anything about cancer, but But basically, he tries to pull these little pieces together, and he decides to focus on the occurrence of leukemia amongst the offspring of the atomic bombs in Japan. And there is data that is coming forth on that. And leukemia is the first cancer that actually occurs. And so basically what happens is that when all is falling apart from this committee, he puts together a first draft of a paper that he then Beadle then gets Ed Lewis to share with his now Baer committee, and he does that in late November, early December of 1956, and he gets comments back from the panel members and so forth, and I have I have whatever is available in from going into all their preserved papers and things of that nature. Now he revises his paper, but in the course of this, he initially focuses on the leukemia issue at Hiroshima, Nagasaki, and then there's a group that has a terrible arthritic condition called ankylosing spondylitis, and they got some very, very high doses of radiation, and they developed also leukemia. And then he looked at some children with very large enlarged thymus glands who would be treated with x-rays to try to shrink the thymus, and some of these developed cancers, and he looked at those. And he also looked at radiologists who he claimed in some papers had 10-fold higher risks of leukemia, and he put all these together, and he developed his version of mathematical modeling, and he claimed that they all followed a linear dose response, and that all these findings somehow reinforced each other from these different types of conditions, all using ionizing radiation, all releasing, all yielding the leukemia. Now, what happened was that when he wrote up a paper on this, but he based his mechanism on mutation, and he went back to the Delta Uphoff the Delta Uphoff papers and her research, and basically justified the mutation mechanism based upon the work of the Manhattan Project. Now, I just have to remind you that in that project, they're not even working with somatic cells. They're working with mature spermatozoa that lack any DNA repair. So he works essentially. He uses the wrong model and the wrong cell within the model to justify a mutation relationship that he thinks is linear, which we know based on the Kasperi work probably wasn't. And so he uses this, and then that's his basis. And so and then he submits a paper to Science. And so now here's a guy who has no experience in any of the relevant areas. I mean, you can start just with, and he's the only author on the paper. It wasn't like he brought in a leukemia expert and a radiation expert and a modeler expert and so forth and so on. No, he does it all himself. And so he basically submits this in, and he gets it accepted into the Journal of Science. And not only does he get it accepted into the Journal of Science, the editor-in-chief of the Journal of Science writes an unbelievably glowing editorial saying that this is like the most extremely important paper, and basically gives him all kinds of publicity. Now, how did he ever get in the Journal of Science? Well, go back to the bottom of this panel here. A panel member of the Baer Committee, his name was Bentley Glass. Bentley was one of the 6 senior editors at Science at the time. And I don't know for sure, but I have a feeling that Bentley Glass would have overseen this and probably provided the vehicle by which Lewis got his paper. And then Bentley Glass probably went to the editor-in-chief and got him to write this grand endorsement, editorial endorsement. And probably my guess is, since the editor-in-chief was an amphibian embryologist who knew nothing about radiation risk assessment or anything, probably Bentley Glass actually even wrote the editorial, but it was just under the name of the under the name of the editor. That's speculation on my part. But at this stage of the game, it makes more sense than the reality that's being shown, I think, to the public. And so this paper had great publicity. It created great momentum for LNT. You won't believe this, but 1 week after it was published in Science, it was discussed in Meet the Press. And then a week after that, he's brought in to testify in front of Congress. And then a week after that, his picture gets a big story, and his picture gets in Life magazine. I had to go and buy the June 10th, 1957 issue just to see it. And yeah, it all happened all at one time. Now, I'd have to say that what happens next here is that it is so happening so fast that he then gets appointed to a big national committee called the National Committee for Radiation Protection and Management, the NCRPM. And that committee is very big still today. And that committee, within a year, makes the very first recommendation to apply the LNT to cancer risk assessment based upon the leukemia studies, and then generalizes it to all other cancers. It all came from the Lewis work. And then the National Academy committees, which still had Muller and the group, they decided to then throw their support into LNT and cancer. And so they just jumped on this particular on this bandwagon. Now, and so when you take a look at this, you find that this this non-entity, this Ed Lewis back in the day, who was not qualified to make judgments, wins the day for LNT, and basically gets LNT established for cancer risk assessment, and it provided the biostatistical basis for how we do risk assessment. It was applied to leukemia, and then generalized to all other tumors, and also applied to chemical carcinogens. And it's what EPA actually picked up on, and the impact, for Lewis continued. He got put on to the Bayer Committee in 1972, and really was extremely influential, assuring that his recommendations would be held. Now the interesting thing, I've gone back and really looked at the Lewis paper, and he failed to analyze the Hiroshima and the Nagasaki data properly. And I mean, he totally, I think, failed in his capacity to understand this and the nature of the dose response. And I'll show you my analysis. And he also failed in how he dealt with the other kinds of endpoints, like the ankylosing spondylitis and the thymus issue and the radiologist, for example. If you go into the ankylosing spondylitis literature, the ones that he cites, and the same for the thymus, if you actually look at the papers that he cites, the authors of the papers that he cites actually state that their findings would not provide reliable estimates, or would not be reliable for providing low dose risk assessment cancer leukemia estimates. They actually say that. But does Lewis actually report that in the Science paper? No, he totally gives it, totally looks past it. Here, the authors, the experts in their area, say do not apply this to low dose cancer risk assessment for the following reasons. Lewis totally ignores that, and he uses that information. Now, he could still use that information, in my opinion, but you have to at least be honest to the reading public to tell them that the authors refused to, they addressed it, and this is why they did, and this is why you disagree with it. He doesn't share that at all. I bring that up, and I show that repeatedly within this. He didn't consider any alternative hypotheses. For example, at this time, a major hypothesis for leukemia was that in about 50% of the cases, a major major infections, viral infections, usually preceded the onset of leukemia. And that still happens in today's world too. They're still trying to figure this out. But basically, he never looked at any of these alternative causation features. There are many other things he didn't look at as well. He just had, a single-minded focus. He knew what the right answer was. He was not really searching for truth, actually. So basically, his use of the mutation data were really shown to be incorrect. He made multiple unverified exposure assumptions that lacked any scientific grounding. He characterized by many as he just made guesses all along the way. He failed to consider the capacity of multiple other factors that could affect disease incidence, that's really because he lacked a broader epidemiologic perspective. And he just, everything that he did smacked of bias. And yet, how did this pass through a science journal peer review? Other than the fact that maybe he had an insider person like Bentley Glass that guided it through. And so the paper, after it was published, was strongly criticized by numerous high-level scientists in the US and Europe. And Lewis did not, that was interesting. Lewis never rebutted, never sent in a letter, never rebutted any of these criticisms. And also very interesting to me was that no member of the radiation genetics community or the Bayer genetics panel came to his defense. Lewis was really left twisting in the wind. And you might think, well, maybe that was the end for Lewis, but it wasn't. Lewis must have had some really good personal skills, because when he got on this big NCRPM panel, he basically brokered a deal with people who thought differently than him and got them to agree to adopt the precautionary principle for cancer risk assessment. And what did that mean here? That really meant that Lewis was a compromise. It meant that Lewis was made to admit that his data were not adequate to support LNT. But it made the people who were critics of him saying that, well, let's be cautious and just assume that LNT is correct. And ultimately, that's where we are today, that Lewis may have lost the scientific battle, but he won the conceptual war, the policy war. And it was really that's at the heart of the how the US EPA works. It's really based upon it's really based upon a precautionary principle. And I want to show you some data here. And these are the data, the actual data that I've plotted from about 12 studies dealing with the atomic bomb induced leukemia. They cover essentially the decade of the 1950s. And I have data that goes out really to the middle of the 1980s. It's not on this slide because I was just comparing for the decade of the 1950s. But this is the decade of Lewis. Now, Lewis shows a linear dose response. And I'll tell you why Lewis gets a linear dose response. But when I look at other data, I don't see that. What we're looking at is the occurrence of leukemia in people with respect to how far they were away from where the bomb was when it landed on Earth, you might say. And where the bomb dropped is called the hypocenter. So it's basically, and so if you're furthest away, like it's really about 1.5 miles to 2 miles away at point number 1. And when you're at point number 4, you are just about there where the bomb is dropping. So the further away you are, of course, is where you might want to be. Now, these people were compared to a control group, national population, or some other type of control group based upon the study that was put together. When you actually look at the data from multiple studies with the same population, but with progressively more examples of cases and deaths that's attributed to leukemia, what you find is that in this zone of about 1.5 miles to 2 miles away from the hypocenter, you see something really weird happen. You find that the occurrence of leukemia is actually less than what is in the control group. It dips down and then it goes sharply up. It's when Lewis presented what he did was it was pretty cute. Lewis followed people out for about, I'm going to say 2,000 meters from the hypo center, and everybody else after that was lumped together. Now, what I found is that it's somewhere between 2,500 to about 3,000 meters away that you see this dip taking place. And Lewis lumped all those, and when you lump them all, you miss this dip below. And I think that the data actually are showing that potential for a hormetic dose response. I actually tried to follow up in the 1970s and the 1980s. I tried to give it a 40-year latency period, and I found the same dip was occurring, and this occurred not just in Hiroshima, but it also happened in Nagasaki. And so this is like a new finding, and that the whole basis is wrong. And all this stuff was out there in the literature, and nobody else put it together. My latest paper shows it. Now, I'd like to kind of summarize all this for you here. So we have key LNT findings in perspective. How did we get into this LNT mess? Well, we got in there, if you go back to the beginning, Muller's gene mutation claim, we know that's now proven totally incorrect. He didn't induce gene mutation. He took a bazooka to these fruit flies and knocked out the gigantic amounts of nucleotides, the gigantic hundreds of thousands of nucleotides. The single hit model is based upon Muller's incorrect interpretation, so it has no credibility from start. Muller's Nobel Prize was basically wrong, and his speech was deceptive. Muller and Stern misrepresented the Manhattan Project findings to promote LNT. You take a look at this Bayer 1 committee panel, it misrepresented the scientific record. Those people, basically, they're praised today. They committed scientific misconduct. That should be, a stamp on their historical record. 1972, the Bayer Committee, we've just showed, based upon the Russell findings and the clarifications by Paul Selby, and then my application back to the data at the time, that these data actually should have supported either a hormetic or a threshold model, and the EPA based their whole thing on a mistake that's out there in the literature that they don't want to act on. So why has LNT succeeded? Well, producing gene mutations was then considered a major advance that totally overwhelmed the field. Contemporary gene mutation criticism was strong, but neither side seemed to be able to win the decision, even though Statler made Muller's life pretty rough. Manhattan Project had lots of influence coming after the war and all. Dropping the bomb scared the world. The Nobel Prize gave Muller and his ideology the platform it needed. The Cold War had above ground testing, and that focused everybody on fallout and concerns. The Rockefeller Foundation and the NAS created a separate genetics panel. They stacked the members. They got the result they wanted. They promoted LNT. The National Academy was full of ideologically oriented people that lied and deceived. Scientific and the toxicology community, well, they absolutely got the LNT question wrong. They gave it a pass. There was self-interest and scientific misconduct. I have letters from the various committee members on the Bayer panels that said that we should try to make sure that we exaggerate the claims because it'll enhance our receipt, it'll get the public agitated and legislators agitated, and it'll bring more grant money to us. I have that written. So even though they might think they're trying to save the world, well, it can save the world, but help my career at the same time. They always talk about follow the money trail, and they always say follow industry. You can go right to an academic, and what is the academic doing? Following the money trail that comes from the funder. I don't really see any conceptual differences, just as much of an issue. All errors and deceptions and mistakes were given a pass. The scientific, toxicology, and regulatory communities failed in their oversight and their review and their leadership. And the bottom line is this, ladies and gentlemen, the entire regulatory programs and public educational activities are based upon such deceptive historical practices. And this is your world. Thank you very much.